In last class, we have seen the one of the most important enzyme, which is performing a function which is needed for the production of recombinant DNA molecule. That enzyme is the restriction endonuclease enzyme. And now this enzyme, why it is called as a restriction endonuclease enzyme? It's because of its character that it only searches the specific recognition sequences and it get bound there, and then it makes the cuts. So there is a restriction for that enzyme to function there. That's why that enzyme is called as a restriction endonuclease enzyme. And it performs the endonuclease activity, means cleaving inside the DNA molecule. So that's why it is called as endonuclease enzyme. So the next enzyme, which is also important in order to make the recombinant DNA molecule, is the DNA ligase enzyme. This enzyme firstly demonstrated by the scientists Mertz and Davis, who firstly demonstrated that the cohesive ends which are produced because of the use of restriction endonuclease enzyme, they can be sealed, they can be rejoined with the help of an enzyme which is present in E. coli, and that enzyme they called as a DNA ligase enzyme. And the function of this DNA ligase enzyme is to rejoin the fragments which are produced because of the restriction endonuclease enzyme. Now this DNA ligase enzyme, it has a property that it seals the single-stranded nicks which is present in the DNA molecule because it has an activity, that is 5' to 3' terminus. When there is a presence of this hydroxyl group at the 3' end of a fragment, this DNA ligase enzyme, it uses this hydroxyl group to rejoin the two separate strands, or to seal the two separate strands. Now here two different type of ligase enzymes they have been known, and which are involved in joining the DNA molecules which are produced after the use of restriction endonuclease enzyme. Or what are the fragments that are generated by using the restriction endonuclease enzymes, they can be rejoined with the help of DNA ligase enzymes. Now there are two types of DNA ligase enzymes depending upon the source from where it is isolated. DNA ligase enzyme which is isolated from E. coli, and DNA ligase enzyme which is isolated from T4-Phage. So there are two sources from where these DNA ligase enzymes they can be used in the recombinant DNA technology. But the main source of this DNA ligase enzyme, it is the T4 bacteriophage, and that DNA ligase enzyme which is isolated or extracted from this T4 bacteriophage, that ligase enzyme is known as T4 DNA ligase. And both of these enzymes, which is either isolated from the E. coli or the T4 bacteriophage, they require some cofactors to function. In case of DNA ligase enzyme which is extracted from E. coli, it requires nicotinamide adenine dinucleotide as a cofactor in a simple manner, NAD+, while T4 DNA ligase, it requires ATP as a cofactor in order to perform their activity. These cofactors, they are essential for these enzymes, and without the presence of these cofactors, this enzyme won't function. And both of these enzymes, they show a common character that it contains amino group on the lysine residue. In both these enzymes, which is isolated from E. coli or T4 bacteriophage, it has a characteristic that it contains amino group on the lysine residue. And this enzyme, it functions differently. Here, these cofactors, they are broken down in order to produce the AMP molecule, adenosine monophosphate molecule, which in turn adenylate the enzyme to form a complex, enzyme AMP complex. And this enzyme AMP complex, it is generated by the breakdown of cofactor into AMP molecule. So there is a formation of a complex which contains enzyme adenosine monophosphate molecule. So these two get combined and they form this complex. And this complex, this enzyme adenosine monophosphate complex, it binds to the NICs, where there is a presence of hydroxyl group at the 3' end, and at the 5' end there is a phosphate group is there. And these which are present in the double-stranded DNA molecule. So this enzyme complex bounds to these two ends, and here this enzyme complex then adenylate the phosphate group which is present on the 5' end of the double-stranded DNA molecule with the hydroxyl group which is present at the 3' end of the strand of that DNA molecule. So it adenylate the phosphate group with the 3' end hydroxyl group, and it results in the formation of phosphodiester linkage between these two ends. 3' end, it get combined with the 5' end with the help of hydroxyl group and the phosphate group, and this reaction is carried out by this enzyme complex. And here there is a liberation of AMP molecule. And how this by using this complex, this DNA ligase enzymes, it rejoins the broken fragments to form the recombinant molecule. Now, these two enzymes, which is isolated from E. coli and T4 bacteriophage, they are also having some another characteristics. Here, T4 ligase DNA ligase enzyme, it has a capacity to join the blunt ends of the DNA fragments. Here, in last class, we have seen that there is a presence of, there are these enzymes, restriction endonuclease enzymes. It produces two types of products by the breakdown of this hydrogen phosphodiester bond between these nitrogen bases of a DNA molecule. It may produce staggered cut or cohesive end product, or it may produce blunt end products. Now here T4 ligase enzyme, T4 DNA ligase enzyme, it has a capacity to join the blunt ends of the DNA fragments. Whereas the DNA ligase enzyme, which is taken from the E. coli, it has ability to rejoin the cohesive ends which is produced by the restriction endonuclease enzyme. Now, these two characters, they are important because by using this, by using this ligase enzyme, we are producing a recombinant product. But after production of that multiple copies of that recombinant product in a host, we have to retrieve the fragment, which is DNA fragment which we require, the gene which we require, that should be recovered. But in this case, the T4 DNA ligase enzyme, it joins the blunt end fragments of the DNA molecule, while the DNA ligase enzyme, which is taken from the E. coli, it has capacity to rejoin the cohesive ends which are produced because of the restriction endonuclease enzyme. But there is a problem is that when we use T4 DNA ligase enzyme to join the blunt end of the DNA fragments, which joins these blunt ends to the double stranded DNA molecule, and a recombinant molecule will be produced. But when we use this enzyme, there is a problem is that whatever the DNA fragment which we are having an interest, that DNA fragments, once it get inserted into the vector, and these blunt end fragments which are inserted into the vector, we are not able to retrieve that fragment when we use DNA ligase enzyme which is extracted from the T4 bacteriophage. But the DNA ligase enzyme which is extracted from the E. coli, which is capable of binding only to the cohesive end products or cohesive end fragments of the DNA molecule, we are easily retrieve that DNA fragment from the cloned vector. So there is a difference between these two ligase enzymes which plays an important role in order to retrieve the DNA fragment which is of our purpose. This is the reaction here. This is DNA ligase enzyme which is having a amino group at the lysine residue. This DNA ligase enzyme it is firstly adenylated by using this cofactor, and this is the enzyme AMP complex which get bound to the phosphate and hydroxyl group which is present at these molecules, and there is a formation of phosphodiester linkage between these two fragments, and there is a release of AMP molecule, and this is how this DNA ligase enzyme rejoins the fragments which is either produced during the replication process or in the recombinant DNA technology. Whatever the fragments that are generated, they are rejoined by this DNA ligase enzymes. And this DNA ligase enzyme, it performs function in recombinant DNA technology as well as in rejoining the Okazaki fragments which are produced due to the discontinuous replication process. Okay, so this is one enzyme which is also having importance in recombinant DNA technology. Then next enzyme is the alkaline phosphatase enzyme. Now, when we treat a plasmid by using a restriction endonuclease enzyme, for example, we have seen in earlier class there is a presence of a EcoR1 endonuclease enzyme, restriction endonuclease enzyme. This restriction endonuclease enzyme it has a ability to produce the cohesive end fragments, which we have earlier seen here. This is the product. This is the EcoR1 enzyme, which is having a recognition sequence. This is the recognition sequence for this EcoR1, and it produces the cohesive end products. Now, these cohesive end products, which are generated because of this EcoR1, this has capacity to rejoin once again to form the same molecule. Now, when same fragment will be rejoined to its original plasmid molecule, then it will be not a recombinant molecule. Suppose if there is an addition of another DNA fragment molecule in between these two ends, in between these two fragments, then it will be the recombinant molecule. But when same fragment will rejoin, then the original plasmid it is reformed. Our interested DNA will not be added in such conditions. So to avoid such conditions, this alkaline phosphatase enzyme it is used. These fragments which are produced because of the use of this restriction endonuclease enzyme. Suppose that EcoR1, this enzyme is used and we have used this enzyme and treated the plasmid, which we are going to use as a vector for the recombinant DNA molecule production. Suppose we treated this plasmid with this EcoR1, it produces cohesive end fragments. Okay, and these cohesive end fragments, this plasmid which is treated with this restriction endonuclease enzyme is then further again treated with this alkaline phosphatase enzyme. Okay, this restriction endonuclease enzyme treated fragments of this plasmid vector, they are further treated with this alkaline phosphatase enzyme. But the DNA fragments, the source DNA, which we have treated again with the restriction endonuclease enzymes, they are not exposed to the alkaline phosphatase enzyme. Here only the vector which we have chosen and treated with the restriction endonuclease enzymes, they are only exposed to this alkaline phosphatase enzyme. Because this alkaline phosphatase enzyme, it has a function that you can see here. This is the nitrogen base which is carrying the hydroxyl group at the 3' end. When we treat this enzyme, when we use this restriction that is alkaline phosphatase enzyme and treated the fragments of that plasmid which are produced by using the restriction endonuclease enzyme. In such condition, this alkaline phosphatase enzyme, it removes out the phosphate group which is present at the 5' You can see here, this is the phosphate group which is present at the 5' of the DNA molecule. When we treat these fragments of plasmid vectors which are produced because of the restriction endonuclease enzyme with this alkaline phosphatase enzyme, this alkaline phosphatase enzyme, it performs a function. It hydrolyzes the phosphate group which is present at the 3' end of the plasmid fragments. So, because of the removal of this phosphate group from the 5' end of these plasmid fragments, these plasmid fragments, they are not able to rejoin its own fragment once again. इथं सिंपल काय होत आहे, प्लाझ्मिड फ्रॅगमेंट जे ट्रीट केलेत आपण ह्या अल्कलाइन फॉस्फेटेज एन्झाइम न, तिथं अल्कलाइन फॉस्फेटेज एन्झाइम काय करतोय, हा फॉस्फेट ग्रुप त्या प्लाझ्मिडच्या फ्रॅगमेंट्स मधून काढून टाकतो. त्यामुळे काय होतं, प्लाझ्मिडचे फ्रॅगमेंट्स पुन्हा रिजॉइन होऊन पुन्हा ओरिजिनल प्लाझ्मिड तयार होत नाही. इथे काय होणार मग, फॉस्फेट ग्रुप कोणत्या फ्रॅगमेंट्स बरोबर अवेलेबल आहे, इट इज ओनली available on the source DNA fragments. So this phosphate group combines with these heteroxyl groups. There is a formation of phosphodiester linkage by using the same DNA ligase enzyme. So we are able to get the recombinant DNA molecule only by treating the plasmid fragments with this alkaline phosphatase enzyme. And this enzymes removes only the phosphate group from the plasmid fragments, because we are only treating the plasmid fragments. We are not treating this DNA source with this alkaline phosphatase enzyme. So phosphatase group is available on the DNA source fragments. They get combined with this hydroxyl group which is present on the fragment of plasmids. And there is a formation of phosphodiester linkage, and this phosphodiester linkage, it is formed because of the DNA ligase enzyme, and we are able to get the recombinant DNA molecule in this case. If we are not treated these plasmid fragments with this molecule, this alkaline phosphatase enzyme, then it may be possibility that these original fragments of these plasmids, they get rejoined once again with the plasmid molecule. Our interested DNA will not be added there, But in such treatment with this alkaline phosphatase enzyme to that of plasmid fragments, then it makes sure that the recombinant DNA molecule will be produced when we treat the plasmid fragments with alkaline phosphatase. Because of the treatment of this alkaline phosphatase enzyme on the plasmid fragments, it removes the phosphate group. So no phosphate group is available on the plasmid fragments to rejoin with its original plasmid fragments once again. But the phosphate group, it is available with the DNA source of our interest. So our interested DNA fragments, they can join with the plasmid fragments and they form the phosphodiesterase linkage. And this joining of plasmid fragments to the DNA source of our interest, it is carried out by this DNA ligase enzyme. So finally we get the higher frequency of production of recombinant DNA molecule when we treat the plasmid fragments with alkaline phosphatase enzyme. This is another enzyme which is needed in the recombinant DNA technology. Then some of the enzymes they are there which are modifying the DNA molecules, and these enzymes, they are also required there. And in this DNA modifying enzyme, the first enzyme is the reverse transcriptase enzyme. And this reverse transcriptase enzyme, it is used to synthesize cDNA library or complementary DNA library by using mRNA as a template. Here, this reverse transcriptase enzyme, we have seen this enzyme in second year in the disease that is the HIV infection. The retroviruses, they carry this reverse transcriptase enzyme and they convert their genetic material, which is the RNA, into the DNA molecule, and from that DNA molecule, RNA will be regenerated again. So this reverse transcriptase enzyme, it is used to produce the cDNA library or complementary DNA library by using the mRNA. Now why we require this complementary DNA library? As we have seen that in case of prokaryotes, the genes, they are present in a continuous manner. But in case of human beings or eukaryotic organisms, the genes are present in two different patterns, intron and exon. Here the exons are the genes while the introns are the non-coding part. Okay, so whenever we are carrying this information and whenever we are using this information in a recombinant DNA technology, then sometimes it may happen that non-coding part may insert it into the plasmid vector. So it's of no use. Whatever the molecule that will be generated after the fusion of this non-coding region with the plasmid vector, then it is not recombinant molecule. It's of no interest. So to avoid that, in case of eukaryotes, firstly, you know that the mRNA, the mature mRNA which is produced in case of eukaryote, it carries the sequence which is complementary to the DNA molecule strand, except there is a presence of uracil in mRNA while thymine is present in the DNA molecule. This is the exception. Okay, so whenever we are going to use the genes from the eukaryotes, we are going to use mRNA, not the gene of that eukaryotic cell, because eukaryotic genes carry coding, non-coding parts in their genetic material. In the DNA molecule, there is a presence of coding and non-coding parts. So to avoid that, we are going to use the mature mRNA. And this mature mRNA, is treated by this reverse transcriptase enzyme. And this reverse transcriptase enzyme converts this mRNA into the DNA molecule, single-stranded DNA molecule. And this is the complementary, means exact copy of that gene. So that's why it is known as complementary DNA library. So this enzyme firstly discovered by the scientist Temin and its co-workers, who discovered that in certain cancers which are caused by the animal viruses, and this animal viruses contains the RNA as a genetic material, and here the transcription of cancerous genes takes place most probably by using a DNA polymerase enzyme which is directed by virus RNA. So they found that here in this cancer causing RNA viruses there is a presence of an enzyme which convert their genetic material into the DNA molecule, and from DNA molecule, this DNA molecule which is generated from that RNA, it is used as a template to synthesize the multiple copies of viral RNA in a cell. And this is used, this information used by the scientists, three scientists, Muzetny and Temin and Baltimore. This information, what information? Conversion of RNA into the DNA molecule with the help of this reverse transcriptase enzyme is used to prepare the cDNA library in case of eukaryotes. Okay, in this case, whatever the gene which has to be taken from the higher organisms, that should be used in the form of mRNA. You know that during the transcription process, a mRNA which is produced, it is the exact copy of DNA molecule, which is complementary to the opposite strand. Except the presence of uracil in mRNA and thymine in DNA. But it is the exact copy. So this reverse transcriptase enzyme, which plays a very important role in order to produce cDNA libraries, and this cDNA library, it is produced only in case of eukaryotes, because in case of eukaryotes the genes are not present in a continuous manner like that of bacterial cell. It is produced in discontinuous manner with coding, non-coding region. So this reverse transcriptase enzyme, it is useful in recombinant DNA technology, where it modifies the mRNA into the DNA molecule. Then another enzyme, it is the DNA polymerase enzyme. Now you know that DNA polymerase enzymes are of three types: first, second, and third. As we have seen this in DNA replication process, where these enzymes, they are performing their function in a different manner. But here, this DNA polymerase enzyme, it performs function in a different manner. Now this DNA polymerase enzymes, they polymerizes the DNA synthesis on the DNA template. Here the cDNA, it is produced by using this DNA polymerase enzyme. Now along with this polymerization reaction which is carried out by this DNA polymerase enzyme, it also carries certain another characteristics. That is 5' to 3' exonuclease activity, 3' to 5' exonuclease activity. These activities, they are also playing really important role in order to remove the primer in case of DNA replication process, in case of proofreading. Now these DNA polymerase enzymes, they have been firstly investigated by the scientist A. Korenbaum and his co-workers in E. coli. And this DNA polymerase, which is discovered by this scientist, it is the DNA polymerase first enzyme. And after that, we have found that there are also two DNA polymerase enzymes are there, DNA polymerase second and DNA polymerase third enzyme. And they are having one thing common between them. That is, they carry out the polymerization reaction only. But they are not able to initiate the polymerization reaction. They require free hydroxyl group presence at the 3' end. Only they can initiate only they can then polymerize the DNA polymer. Now this DNA polymerase first enzyme, it is required here, is required in DNA, recombinant DNA technology. And this polymerase enzyme has a molecular weight 1,09,000, which is made up of a single polypeptide chain, which is having about 1,000 amino acid residues. And this enzyme has a polymerization activity, means it can add the fresh nucleotides to the 3' end, which carries the hydroxyl group with them. So it adds the fresh nucleotides to the hydroxyl group which is present at the 3' end of the fragment. Along with this, the DNA polymerase first enzyme, it also has two different activities, 3' to 5' exonuclease activity, where it hydrolyzing the single nucleotides residue from the 3' end of the DNA fragment. Similarly, it also has 5' to 3' exonuclease activity, where it hydrolyzes the single nucleotide residue from the 5' end of the DNA fragment. You can see here, this is the 3' to 5' end exonuclease activity, where the single nucleotides will be removed by 3' end. While similarly, this DNA polymerase first enzyme, it also has another activity, that is 5' to 3' exonuclease activity, where it removes the single nucleotides from the 5' end of the fragment. Now this DNA polymerase first enzyme, it is sensitive to the digestion by the protease enzyme. It can be easily degraded by the protease enzyme. When this DNA polymerase first enzyme, it is degraded by the protease enzyme, it produces a large fragment which carries the polymerase and 3' to 5' exonuclease activity. Okay, when we treat this DNA polymerase first enzyme with the protease enzyme, this DNA polymerase enzyme, it is broken down into two different fragments. One large fragment, it contains the polymerase activity and 3' to 5' exonuclease activity, but it lacks the activity of 5' to 3' exonuclease. And this portion of polymerase first, that is the last subunit of that polymerase enzyme, it is known as a clinopolymerase. And this performs the activity, it carries out the polymerization of the fragments. You can see here, this is the 3' end. When we treat this enzyme, treat this fragment with this enzyme, clinopolymerase, it adds the nucleotides according to the template at the 3' end of the DNA fragment. So we can able to get the intact DNA fragments by using this DNA polymerase first enzyme. And this is also DNA modifying enzyme. Then next is another enzyme is there, S1 nuclease enzyme. This S1 nuclease enzyme, it has a capacity. Now we have seen that when we treat that particular fragment with the restriction endonuclease enzyme, it either produces blunt ends or a cohesive end products. Now, in some cases, the cohesive end products were not needed. We require blunt end products. Then, in such condition, in order to produce the blunt end product, we require this S1 nuclease enzyme. The characteristic of this S1 nuclease enzyme, it degrades the single-stranded protrusions of the double-stranded. Here it can easily break down the overhang. This is the, you can see here, it is the overhang. You can see here, this is the cohesive ends or staggered cuts. Now this S1 nuclease enzyme converts this staggered cut into the blunt end products. You can see here. After the treatment of this fragment with this S1 nuclease enzyme, it removes this TTAA fragment from that molecule. You can see here. This is the blunt end products which are generated by the treatment of this fragment with the S1 nuclease molecule. So this S1 nuclease enzyme, it is required for the conversion of cohesive ends into the blunt end products.